Hey guys, so today I'm going to be doing a quick video on apoptosis, also known as programmed cell death. We're going to run through the extrinsic and intrinsic pathways and go through some of the key players. So, what happens in apoptosis? Well, a signal can either come from outside or from the extrinsic pathway, or from inside from the intrinsic pathway. We're going to be going through each of these step by step, and then I'm going to do a quick summary. Okay, so extrinsic pathway. When you think of extrinsic, you're going to think of Fas. Okay, so Fas is a receptor found in our cell membranes, and when the ligand for Fas is bound, this will activate its death domain. So it will activate FAD, or the Fas-associated death domain, which is attached to the Fas receptor, or the Fas-R. This Fas-associated death domain will then activate the death-inducible signaling complex, which is going to activate our caspases. So, caspases are key proteases involved in the apoptotic pathway, and when they are activated, caspase 8 is going to go ahead and cleave and activate caspase 3, and caspase 10 is going to activate BID, which then goes to act on the intrinsic pathway, and we'll cover that in just a second. So here, DISC is going to activate caspase 8 then activates caspase 3, and our main player in apoptosis, whether you're talking extrinsic or intrinsic, is caspase 3. This guy is going to activate nucleases, which acts on the nucleus, obviously, and causes cell death. So what do we have going on with the side pathway drawn over here? This is perforin and granzyme. So these are released in little granules by either natural killer cells or cytotoxic T cells, and these granules, when released, perforin creates a hole in the cell membrane, and granzyme is going to go inside the cell and activate caspase 8 and caspase 3. So these guys also contribute to the extrinsic pathway. The intrinsic pathway, on the other hand, is exactly what it sounds like. The signal comes from inside the cell. So here we have DNA damage as our main cause for the intrinsic pathway activation. This is going to activate ATM. So this ATM protein is a kinase, which you might remember from the S checkpoint in cell cycle arrest. So the cell, when it checks for DNA damage during replication, ATM is involved in the S checkpoint. And ATM is a kinase, so it's going to phosphorylate and activate P53. So P53, our main tumor suppressor gene. P53 has a lot of actions, so it's going to activate BCL2 family proteins, P21, which is involved in cell cycle arrest, and IGF-BP3, which I don't think is that important, so we're just going to skip over that one. Okay, so P53 is then going to go and inhibit BCL2. Let's talk about BCL2 for a second because it's an important pathway to know. So BCL2 is regularly bound to BAX and BAC. These BAX and BAC proteins are found on the outer mitochondrial membrane, and they are regularly inhibited by BCL2. But when P53 is activated by ATM, it's going to go ahead and phosphorylate BCL2, releasing it from BAX and BAC. Now BAX and BAC can go ahead and create a channel inside the outer mitochondrial membrane. So now cytochrome C, which is in the intramembrane space of the mitochondria, can escape. Once cytochrome C escapes the mitochondria, it can go ahead and bind to APAF1 and caspase 9. And these three proteins together are going to form something called an apoptosome. This apoptosome is very important because, again, it will activate caspase 3 and a bunch of other caspases and cause cell death, which is our main point of apoptosis. Okay, so P53 is going to bind BCL2, cause it to be released from BAX and BAC, and cause cell death. Okay, so this might be a little confusing for you because here we have BCL2, and here we have the BCL2 family proteins. These BCL2 family proteins I've outlined in blue circles. They are actually pro-apoptotic. BCL2 itself is anti-apoptotic. So when BCL2 is bound and active, it's going to not let BAX and BAC release cytochrome C. So remember that the BCL2 family is pro-apoptotic, and BCL2 itself is anti-apoptotic. So here we have caspase 10 activated from the extrinsic pathway. This is going to activate BID, which is a BCL2 family protein, and that's going to phosphorylate BCL2 and cause it to be released from back. Again, the same ththing happens with P53's activation of BAD, and that does a very similar process to BID. So it's going to release BCL2 from back. Okay, so I hope this video helped get you caught up on apoptosis and outline the general steps. All right, I'll see you guys in the next video.